Hi, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Dr. Donn Van Deren, and I'm a postdoc at the University of Utah. I will be moderating today with Dr. Lydia Grmai, a postdoc at the University of Pittsburgh. Now, both of us are quite excited to highlight the work of our outstanding postdoctoral members. And today, Dr. Daniel Aldea from the University of Pennsylvania and Dr. Harini Iyer from Stanford University will share their research. Now, each speaker will give a 20-minute talk, followed by 10 minutes of questions and answers. Now, if you have questions, please enter your questions in the Zoom Q&A box, and they will be answered at the end of the speaker's talk. And so moving forward, I'm elated to have the opportunity to introduce our first speaker, Dr. Daniel Aldea. Dr. Aldea has quite a unique background. His master's thesis work under Dr. Silvian Marcellini at the Universidad de Concepción consisted of examining genetic changes leading to the evolution of the vertebrate bone matrix. Dr. Aldea was awarded the prestigious Beca Scholarship from the Chilean Ministry of Education to perform his doctoral studies in France. And his PhD work under mentorships of Dr. Stephanie Bertrand and Hector Escriva at the Pierre and Marie Curie University in Paris entailed uncovering the origin of the vertebrate head through studies in lancelets. He is currently a postdoc in the laboratory of Dr. Yana Kambarov, where his training encapsulates the role of non-coding elements in the development and evolution of human-derived skin traits. Now, without further ado, allow me to give the platform to Dr. Aldea, where he will present his current research on how distinct gene regulatory modules inform the evolution of eccrine gland density in humans. Take it away, Dr. Aldea.
All right. Thank you, Don, for the introduction, and good afternoon, everyone. And good afternoon, everyone. I just want to say thank you. I'm very grateful to SDB for giving me the opportunity to share my work here today. As Don said, my name is Daniel Aldea. I'm an evolutionary and developmental biologist, currently a postdoc in the Yana Kambarov lab at the Department of Genetics here at the University of Pennsylvania. And today, I'm very excited to talk to you about how the cis regulatory machinery of mammals, they have evolved to modify one unique and single trait that is the eccrine sweat gland density. So humans are different from any other primate and actually any other mammals out there in the single mechanism that we sweat to cool off. So what makes this possible is that our skin is fully populated of a type of exocrine gland called eccrine sweat glands, or eccrine glands that basically are composed of a coil and a tube. In response to high core temperature, whether you are performing physical activities or you have like a feverish period, this gland will pump up water onto the surface of the skin. This water will evaporate because of the heat of your body. And this is the mechanism that allow you to cool down your body temperature. So our thermoregulatory capacity is so efficient because we have massively increased the number of eccrine glands in our body. So just to put it in context, human, if you inspect the skin of different primates, you will see that human has on average 10 times more eccrine gland compared to other primates, so such as chimp or macaque. So again, sweating is the main mechanism of thermoregulation in humans. And indeed, if you have any problem with this gland, if you lose this gland, this can lead to overheating or heat stroke in our species can be lethal. So because of the important role of eccrine gland in humans, the question that have been addressing in the past few years is how humans evolved their high eccrine sweat gland density, what is the genetic mechanisms underlying the evolution of these human adaptive traits. So when we thought about this question, we thought about development, because it is known It is known that evolution tends to work by modulating, by tweaking a developmental program to modify a trait. So what we know about eccrine gland development is mostly from work done in mice. So we know that there is a transcription factor called NGRAIL1 or N1 that plays a critical or plays a critical role to promote the formation of eccrine gland in the skin in the skin. So a difference of humans, that as I said, we have eccrine gland in all over our body. In mice, eccrine glands are restricted to the ventral area of their paw, or what is known the volar skin. So here you have an epidermal preparation of an adult mouse paw skin. So this tissue, we stain this tissue to better visualize the ectodermal organ that there is in. So in this zoom area, you can observe that this is an eccrine gland, an adult eccrine gland. This is the duct of an adult eccrine gland in mice. So we also know from work done in mice that increased level of N1 in the skin promote the formation of more eccrine glands. So here you can see a cross-section of an in situ hybridization at a time point where eccrine glands are developing. So that is perinatal day P2.5 or perinatal day 2.5 or P2.5. So you can see that N1 is expressed through the deepest layer of the ectoderm and is upregulated in the plaque that will give rise to the eccrine gland. So very importantly, we also know that in during human skin development, there is an upregulation of N1 that correlate with the onset of eccrine gland. So that's why we thought that human might have evolved more eccrine gland by modulating the level of expression of N1. So you can modulate the level of expression of any given gene through enhancer, that this non-coding regulatory sequence. And we have recently performed a screening looking for enhancer, N1 enhancer, that are active during skin development. So using human skin cells and performing functional analysis in mice, we found that there is an N1 enhancer that has been mutated over and over and over again to keep increasing his activity, specifically on the human lineage. So this results in a more potent enhancer that can activate more N1, you have more NGRAIL, and therefore you have more eccrine gland in the human lineage. So we call this enhancer ECE18, that stands for N1 candidate enhancer 18. So this was the first study that actually showed a genetic basis of one of the most distinguished human adaptive trait, that is, or a large number of eccrine gland. But you know, what is curious about this element is that only act as an enhancer in a subset of primates. So outside of primates, when we look in mice, when we delete this enhancer using CRISPR-Cas9, we observe that this element doesn't act as an enhancer because we didn't observe changes in the expression of N1 or changes in the number of eccrine gland in mice. But we know that mouse or other mammals, they make eccrine glands and they use NGRAIL to do so. So this, so we can see two scenarios of why we don't see that EC18 is not regulating N1. So the scenarios is that either the mouse or other mammals, they have evolved a completely different mechanism to regulate N1, or it can be also that this mechanism is conserved across placental. So these are the questions that I'm going to address today, and the question that I'm going to talk to you today is there any other N1 enhancer controlling eccrine gland formation? Is this element shared or lineage specific? And what is the relationship of this element with ECE18? So to address this question, we came back to our original screening, in which we look for a non-coding region or enhancer that were within the topologically associated domain of N1 or N1 TAT. You can see the N1 TAT is around 800 kilobases. So here you can see N1, the coding region. Also, this element were, as I mentioned, were active in the during skin development, and this element were evolutionary conserved in terms of sequence. So this is the element that I just mentioned to you, is the 18 that is located around 400 kilobases downstream of N1, And today I'm going to talk about this other element that is called ECE20. So what we know about this element, we know that ECE20 can drive reporter expression in the N1 domain during eccrine gland development. So we determined this using a transit transgenic mice. So briefly what we did was to use a lentivirus approach to see this. So basically we clone ECE20, mice ECE20 upstream of a minimal promoter and upstream of a reporter gene, in this case GFP. We pack this into lentivirus, we infect the mice using ultrasound guide needle at E9. At E9, you have just one monolayer of ectoderm, so this ectoderm will give rise to all these ectodermal organs, including eccrine glands. And we will expect to get several events of transgenesis. And then we look the reported activity of this element within the N1 domain. So here you see the N1 domain at P2.5, the time where eccrine gland are developing. You can see this developing eccrine gland as well here. So we found that the mouse ECE20 can drive a reporter expression in mice. We use HRP DAP immunostaining to detect GFP. So that's why a positive you will see as a dark brown cell, as you can observe here. So you can observe this this positive cell here, and also in this developing eccrine glands. So also we undertook a similar approach to see if the ability of the human ECE20 to drive a reporter expression in basal keratinocyte cells. We got similar results. So we know now that ECE20 is an element that can drive a reporter expression in basal cells in the tissue basal keratinocyte cell, that is the tissue that we get. But that is matter. Does this element actually control N1 expression? And to address this question, what we did was to engineer the mouse genome. So what we did was to delete this enhancer using CRISPR-Cas9 to create a mouse that we call mouse ECE20 knockout. This enhancer that we need. So then we check the N1 expression in the tissue that we care, that is when the eccrine gland are forming. And we observe that there is a drop in the N1 expression. So you can observe here, this is the full chain of N1 expression. And you can see this drop of the N1 expression when we compare wild type and the mouse ECE20 knockout. So next we ask if this drop in the N1 expression is due to a cis or a trans effect. Because it can be that actually this enhancer is controlling some other transgene, and this transgene is controlling N1, or it really can be that the mouse ECE20 is controlling in cis, in the same allele, the expression of N. So to discriminate between these two scenarios, between these three options, what we do was to perform an allelic discrimination assay. So basically, we breed two different strain of mice, and in doing so, we can measure how much N1 is coming from one allele or to other, and one of them will be a wild type, and the other one will be carrying the mouse ECE20 deletion. So we observe that there is an skew on the N1 allelic ratio in the mice that they have the deletion. So this is really telling us that the effect that we observe, the drop in the N1 expression, is in cis. So it's really, this is an enhancer, a developmental enhancer of N1. So great, we have this element that we call ECE20, that is a developmental enhancer, but what about the phenotype? Is this element actually affecting the phenotype? So to address this question, what we did was to score the number of eccrine gland in adult mouse. So as I showed you in the beginning, these are epidermal preparation of adult mouse foreskin. So you can see these are the footpad of the mouse. A footpad is pretty much like the footpad of your dog or your cat at home. And we score this area here, this central area here, that contain eccrine glands. So again, these are the eccrine glands. These are duct of the eccrine gland, as you can observe here. So we score the two different genotypes, and we observe a drastic drop in the number of eccrine gland when we delete this enhancer, as you can observe here, was quantified in this plot when we compare wild type and mouse ECE20. So this correlate with what we observe at the level of RNA expression. And then we ask if actually this element can, what is the relationship of ECE20 with ECE18? So as I mentioned at the beginning, when we delete ECE18 in the mouse genome, we didn't observe any effect on the N1 expression, nor in the number of eccrine glands. So we really wonder if that what we observed there is because ECE20 was compensated in somehow these other enhancers, so there was some relationship between them. So to address this question, what we did was to engineer another mouse. So we delete both enhancer in this mouse, and we call this element in this mouse mouse ECE18 knockout and mouse ECE20 knockout. So as you can observe here in this, we quantify the same area, the central area, the number of eccrine gland, and you can observe here that for both cases, the single and the double deletion, we observe a drop in the number of eccrine gland, great, compared to wild type. But then when we compare both, we observe that there is no significant difference between these two animals. So this is telling us that these enhancers are not acting redundantly to control eccrine gland number. So just to sum up this part, what I just tell you so far is that we discovered this new element that we call ECE20. This element has a shared enhancer activity across placental mammals. This element is required for N1 to control its activity. This element can modulate the number of eccrine gland. And this really contrasts with what we have found recently, or we have found in the past, that this that is ECE18, that this ECE18 is a lineage-specific enhancer. So we have here like a shared module, a shared enhancer, and also a lineage-specific enhancer. But this raised a question, which is why we have these two different types of enhancer? Why we have a shared one and a lineage-specific just for primates? Why we bother in having these two kind of like elements? And we really bother because we have recently shown that as I mentioned at the beginning, that the human ECE18 has been mutated over and over and over and over again through evolution and in the lineage of humans. Indeed, this element overlap a human accelerated region. And we found that in this element, there are 10 human-derived nucleotides relative to ancestral A. So that means at any of these 10 positions, if the human has a C, for instance, the other ancestral A, they will be similar. And for instance, here they have a T. So we can quantify the enhancer activity of this element. We did that for the case of ECE18 and the homolog, and we observed that the human ECE18 has greater enhancer activity compared to chimp. And then we mutate each of these mutations back to the ancestral A to observe which one of them has an effect on the enhancer activity. However, we did not observe much of an effect in each of them. But when we mutate all of them together back to the ancestral A, we observe this drastic drop on the enhancer activity. So this is really telling us that is the combination of all of them. They are epistatic interaction between this mutation, and this is what gives to the human ECE18 this greater enhancer activity. So this is really unusual. This is really unusual in evolution, that evolution is targeting the same component over and over and over again to start gaining like an enhanced activity. And another aspect of this element is that it's super specific. We found that ECE18 is a skin-specific enhancer because we have what we did also previously is that we replaced the mouse ECE18 by the human ECE18 at the same genomic position using CRISPR-Cas9 to make this mouse, to humanize a mouse. And we observed that when we did that, we observed an increase in the N1 expression. And this correlates with the increased number of eccrine gland in this humanized mouse. But we also know that in gray is a developmental gene that is important for the formation of other tissues. For example, play a key role for the formation of the mean-hand brain, for the formation of the boundary, for the regionalization of the brain. So here you can see this is an embryo at E10.5 of mice. You can see the expression of N1 in this in situ hybridization here. So we check what happened with expression of N1 in these humanized mice. We did not observe much of a change, and this really contrasts with what we discovered with the case of ECE20. Because for the case of ECE20, we observed that this element, as I mentioned before, this element can control the expression of N1 in a skin-specific manner, but also can control the expression of N1 in the brain. As you can observe here, in this, there is an skew on the N1 allelic ratio when we delete the mouse ECE20 knockout. So we know that, and it's very well described, the role of N1 or how the midhindbrain is formed during development. It's a work that started already back in the 90s, 95. And here you can see, so we really know, we know very well what is the gene regulatory network that is controlling the formation of the midhindbrain. So we know that Grmai is at the core of this gene regulatory network. So that's why we pick we select some of these genes to see if they are affected upon the deletion of ECE20. And as you can observe here in this qPCR analysis, this is the fold chain of these genes. You can observe that upon ECE20 deletion, we observe a downregulation of this gene. So the deletion of ECE20 is really affecting the biology of the brain and also the biology of the skin.
One minute.
Got it. Perfect. So we found that the ECE18 is a pleiotropic enhancer at contrast of ECE20 is a pleiotropic enhancer at contrast of ECE18. So in just 1 minute, I just want to wrap up everything what I told you today. Today, what we propose is that the different properties of N1 enhancer is what shape how a mammalian eccrine gland evolved. So on one side, we have this element called ECE20 that is conserved, that have this conserved and a shared activity across placental mammals. This element, and there are some constraints for this element, because the cost of mutate this element is super high. You don't want to mutate something that is acting in two different tissues. And this really favor the evolution of another element that is ECE18, that as I mentioned, and as we showed before, this element has been specifically mutated over and over again in the human lineage. And this is what increased the N1 expression in the N1 expression in the skin, and therefore has contributed to our high eccrine cell gland density. So what we propose here is not only a genetic basis for how eccrine gland density have evolved, but also we learn more about the logic of evolution. So we are really grasping what is the logic of evolution. And with this, I just want to say thank you to the Canberra lab, to Janice, have been very supportive. Verena, Heather, Reiko, everyone in the lab is very awesome. And also the collaborators, the funding sources, and again to the SDB Society for giving me the opportunity to present my work here today. And yes, now I'll be happy to answer any question if you have.
All right. Thank you, Dr. Aldea, for the excellent thought-provoking talk. Honestly, I'm pretty naive to sweat glands. So I've really learned a lot with sweat gland formation. Thank you. Already we have a number of questions, so I'll just go ahead and start reading them off. Let's see. So this is a question from Ruth Williams. Oh, here we go. So this is a question from Ruth Williams. Very interesting work. Did you determine if your enhancer deletion causes changes in the 3D chromatin structure, which could be responsible for the observed phenotype indirectly of enhancer function per se?
We did not check for a 3D chromatin structure for when we delete this element, which could be the responsibility of the observed phenotype. But we show that that by the allelic discrimination assay, as I mentioned, show really like that there is a direct control of this element with the promoter of N1. So yeah, this yeah, I hope that this answered the question. If you want a clarification, or I can.
Okay, great. We have a question from Jessica. Within human, how much of the variation in sweating is due to variance in eccrine gland numbers versus activity? Is there variation in engrailed expression across individual humans that contributes to individual variation in sweating?
Yeah, within human, how much variance to the settings? Yeah, we know that there is variation in the sweating of this within humans, in a number versus activity. Much of the variations.
Okay.
Yeah, we know that there is variation within humans versus activity. It's true there are some sweat glands that are not active. Is there variation in engrailed expression across individual? So that's yeah, that is a great question. We don't know. Actually, when we check for ECE18, there is no changes, or there is no SNP. It's human-specific SNP, at least in this fragment, ECE18, across humans. We don't know about other regions, but that is a very interesting question. And at least for the case of ECE18 and for the case of ECE20, there is no human variation in these two elements. Human-specific variation across different populations.
Great answer. Okay, so there's actually two questions from Kyle. I'll start reading the first one. Thanks, Daniel. Really interesting presentation. Two questions. First, if I understand correctly, you showed that individual deletion of ECE18 or ECE20 enhancers led to a partial but not complete loss of EN1 expression. Do you think they are redundant shadow enhancers, and that combined deletion of both would abolish eccrine glands?
Yeah, yeah, maybe it was my very well, but yeah, I showed that the deletion of ECE18 or what the deletion of ECE18 in mouse, we did not observe a change in the N1 expression or the number. With ECE20, this is what we showed, that there is a change in N1 expression and the number, and that led to a partial but not complete loss in N1 expression. We are right in that, but just for the case of ECE20. Do you think that they are redundant with shadow enhancer and that the combined deletion of both will abolish eccrine gland? Well, this is what we did in sorry if I didn't explain it very well. This is what we did in the middle of the presentation. We did both enhancer, ECE18 and ECE20, and we did not observe the total. We didn't like get rid of all of these eccrine glands, just the number that we got, the reduction of the number when was similar when we just delete the single ECE20. So yeah, so yeah, I hope that answers. So deletion of ECE18 again, deletion of ECE18 alone doesn't cause an effect on the mice. With ECE18, yes, cause an effect on the number of eccrine glands. And we delete both. We observe just the effect that was produced by the deletion of ECE20. And yeah, this is the experiment that I show in the middle of the presentation.
All right. And, great. And then the second question from Kyle is, second, a naive question. What is the embryonic cell of origin of eccrine glands? Do they come from surface ectoderm? Do epidermal placodes come from surface ectoderm? Or
yeah.
basal stem cell keratinocytes?
Yeah, yeah, that's a good question. Well, the embryonic origin of eccrine gland is cell come from the ectoderm. The ectoderm start to proliferate, and it's actually from the basal keratinocyte cells, as you mentioned there, is what is the progenitor of the eccrine glands in mammals. So yeah, it's come from the basal cell keratinocyte cells. That is the embryonic origin. And yeah. Okay. So ectoderm, and this ectoderm will like proliferate. You will have different layers, and the basal keratinocyte cells is what will produce an eccrine sweat glands.
Excellent, excellent. There is a question from Marcia. The shape of the eccrine glands in your ECE20 knockout look different than the wild type eccrine glands. Did you look at morphology or check their functionality?
Yeah, that's a great question. We did not look at the morphology in the adults or nor to the functionality. So we don't know if these glands are actually they actually have a normal role in the adults. As far as we can tell, they look normal. Mice they look normal. They don't really need like a eccrine gland to thermoregulate. They are not like humans. That humans really need eccrine gland to thermoregulate the body. In mice and generally in most mammals, the role of eccrine gland is just to like get a little bit of grip when they have to run. So they really are important for what is called a fight or flight. Yeah, we haven't checked that, and we have no reason to believe that they are not functional, but it's something that we can test, yes. And about the morphology, yeah, as I mentioned, we haven't explored that, but to us they look pretty much similar. It's just that the number that is changed between a knockout and a wild-type.
Perfect. We'll just probably have time for one more question. I don't see any more questions on the dashboard, so I'll go ahead and ask. I have a few questions.
Yeah,
they're probably more naive questions. But to start off with, for eccrine glands in humans, the sweat glands, is there a higher density in certain areas of the body? If so, could you comment on perhaps like evolutionary advantage to having sweat glands, like maybe concentrate in certain areas of the body, or maybe they're just distributed evenly?
Yeah, that is actually a very great question. There are differences across the body in the terms of the number of eccrine glands in humans. So the area that we have, one of the areas that we have most eccrine glands are the palm or the sole. And then you will have the forehead that we have a lot of eccrine sweat glands. We believe that eccrine glands, which you have many here again, because this is an ancestral role of eccrine glands. So we just have eccrine glands here in our hands. And as you mentioned, there might be some evolutionary advantage to have a greater number surrounding our head, like in the forehead, and here in the forehead, because you know, when you run and you really like hit cool down your body temperature, this area get very heat, right? And so it's a very important area that you want to like keep like cool, like because you have your brain. So these are the area that you have more like here, the forehead, and then you have the back and these areas. Yeah, but there are some regional differences in humans.
All right. Well, this concludes the first half of our seminar. Now, if anyone has any further questions to ask Dr. Aldea, please type your questions in the Q&A box, and he'll be able to respond to them during the second half of the seminar. Again, thank you, Dr. Aldea, for sharing your vibrant research. And with that, I will now pass the mic to Dr. Lydia Grmai.
Great. Thank you so much, Donn. And thanks again, Daniel, for that awesome talk. So our second speaker today is Harini Iyer, who is a postdoc at Stanford University. She completed her PhD at the University of Illinois, Urbana-Champaign in the lab of Dr. Philip Newmark, where she studied the male germline in parasitic flatworms. And she's now working in the lab of William Talbot at Stanford, studying the role of endolysosomes in microglia. She has been awarded several prestigious fellowships during her postdoc, including a postdoctoral fellowship from the American Heart Association, Dean's Postdoctoral Fellowship, as well as a Developmental Project Award through Stanford's Alzheimer's Disease Research Center, which is typically actually awarded to faculty members. So that is extra impressive. And today she will be sharing with us her work on the chemotaxis and function of microglia in zebrafish. So without further ado, I will turn this over to Harini.
Thank you, Lydia, for that wonderful introduction. And thank you everyone who is on this call for joining us live for this webinar. I'm also deeply grateful to the SDB for this wonderful opportunity to share a small part of the research that I have performed as a postdoc in Will Talbot's lab at Stanford University. And since I'm currently on the job market, I'm going to take this opportunity to share an overview of the research projects that I envision for my future lab. The overarching question that defines my research interests is, how are complex biological systems built? How are they maintained? And how can they ultimately fail due to aging or disease? As a developmental biologist, I'm primarily interested in signaling pathways that instruct and orchestrate the formation of these complex tissues. Simultaneously, I believe that understanding the fundamental principles that dictate how a system is built is in turn essential for us to understand how a system is maintained and how it responds to external forces such as environmental stress. And going one step further, I also believe that a better understanding of the basic principles of development and maintenance are in turn essential for us to decipher how these intricately built biological systems can break down in the context of aging or disease. The complex biological system that I'm particularly interested in is the neuroimmune system, which brings together cells from two very different tissues of origin, the nervous system and the immune system, to essentially drive the supercomputer that is our brain. And whereas most people are drawn to the nervous system half of this equation, I'm captivated by these immune cells called microglia, which are specialized brain-resident macrophages. The importance of microglia becomes immediately obvious when one realizes that whereas the rest of our body has the entire repertoire of both innate and adaptive immune cells protecting it, the brain is an immune-privileged environment, which means that under normal, healthy conditions, there should be no other immune cells in the brain with the exception of microglia. As a result, microglia perform several functions for the well-being of the brain and exist in multiple states. Resting microglia are highly ramified, and they're constantly surveilling their environment for signs of injury or invasion. In addition, resting microglia also perform pruning functions, synaptic pruning, as well as provide metabolic support. When activated, such as in response to pathogens or presence of cellular debris, microglia retract their processes, becoming more amoeboid, and they proceed to chew up and digest the debris. In the healthy brain, microglia go back and forth between these states. In the context of certain neurodegenerative diseases, however, microglia can become constitutively and/or aberrantly activated. In Alzheimer's disease, for instance, this constitutive activation leads to prolonged inflammation, which in turn leads to the loss of viable neurons. Now, given the overarching question that drives my research interests, perhaps you can appreciate, just as I did, that microglia are particularly suited to ask fundamental biological questions about signaling pathways in development, in response to stress, and in disease, which in this case happens to be neurodegenerative disorders. Now, consistent with this very important role of microglia in neurodegenerative diseases, it has been shown that neurodegeneration-associated genes are highly enriched in microglia. Importantly, many groups have shown that microglia that are cultured in vitro rapidly lose their identity and show altered expression of disease-associated genes. In this example, you can see that among genes that are highly expressed in microglia and enriched in brain diseases, about half are significantly up or downregulated when microglia are cultured in vitro. And this differential expression is particularly stark when it comes to genes associated with Alzheimer's disease risk variants, which are highly differentially expressed between human microglia that are cultured ex vivo versus in vitro. So in light of these observations, I will be using in my independent lab the vertebrate model organism zebrafish to study the functions of microglia. I know that this audience does not really require an introduction to zebrafish, but what I will do here is highlight the advantages of zebrafish that are particularly relevant for my research. Zebrafish show rapid external development. For example, here you can see that a fertilized egg goes from a one-cell stage to a fully formed embryo, and all of this happens in 24 hours and in a dish. What this means for my research is that I can observe the distinct stages of development of the neuroimmune system, which isn't really possible in an organism that develops in utero. Zebrafish also produce a large number of eggs, which makes large-scale genetic screens feasible, as I will discuss at the end of my talk. And finally, and perhaps most importantly, the transparency of the zebrafish larvae allows us to perform live imaging and observe in vivo processes. And here you can appreciate macrophages circulating in the zebrafish larva. And throughout the course of my talk, I will be highlighting these various advantages of zebrafish for my project, particularly emphasizing the intimate link between microglia functions and lysosomal pathways. If you take a step back and think about the various functions of microglia, whether it's clearance of apoptotic debris or tumor cells or even misfolded proteins, essentially all of these functions have to culminate in lysosomes for degradation or recycling. And while I think we all intuitively understand that as professional phagocytic cells, of course, microglia rely on lysosomes for their clearance functions, I think we're just beginning to appreciate how aberrant lysosomal activity can in turn contribute to microglia development, their functions, and even some of the pathological states of microglia that I talked about earlier. So narrowing down the focus of this pretty broad question that I asked at the beginning about how the complex neuroimmune system is built, how it's maintained, and how it can fail in disease, members of my independent lab will specifically ask the question, how does aberrant lysosomal activity contribute to the disruption of microglial function during development, in stress, or in disease? And with this brief background, today I will discuss I will give the talk in 3 parts. I will first share a small part of my postdoctoral research where I will ask what is the role of lysosomal signaling in the development of microglia. In the next 2 parts of my talk, I will briefly discuss my future plans. And in the first part, I will show you how I intend to follow up on the observations from the first part of my talk in my own lab to ask how are lysosomal pathways activated in response to stress. And towards the end, I will talk about, I'm sorry, something went wrong with the transition. And towards the end, I will talk about how we can harness the advantages of zebrafish to better understand neurodegeneration. And I think once again, you can appreciate that this outline is consistent with my theme of wanting to understand lysosomal pathways in development, in environmental stress, and in neurodegeneration. So the preliminary data for the first part of my talk was generated before I even joined my postdoctoral lab, where a forward genetic screen revealed that the lysosomal GTPase RAG-A is essential for microglia. Briefly, Kim, a previous graduate student in the lab, showed that whereas wild-type animals have many ramified microglia in their dorsal midbrain of zebrafish that is shown here, RAG-A mutants have significantly fewer microglia, and these are highly amoeboid. However, and you might remember that this is indicative of possibly aberrant activation of these cells. However, the developmental timing and mechanism of the RAG-A mutant phenotype remained unexamined. So when I joined the lab, I started with the earliest step of microglia formation, their specification. Embryonic macrophages that are specified here in the anterior lateral plate mesoderm at around 22 hours post-fertilization migrate through the yolk to colonize the brain and form microglia. And for the rest of my talk, we will be looking at microglia in the dorsal midbrain that is shown here. And I will be labeling both microglia and macrophages using a transgenic line that drives GFP under the macrophage lineage-specific MPEG promoter. In wild-type animals, you can see that the number of microglia steadily increases over developmental time, which is what we anticipate. In RAG-A mutants, this number initially goes up slightly, however, remains at a steady low number over developmental time. And if you compare the left and right panels, I think you can appreciate that the biggest difference that we see is between 2.5 and 3 days post-fertilization. And I mentioned earlier the advantages of zebrafish for performing live in vivo time-lapse imaging. So to determine the cellular mechanism that underlies the RAG-A mutant phenotype, I set up time-lapse movies between 2.5 and 3 days post-fertilization. And before I start the movies, I want to orient you to these images. So once again, we're looking at the dorsal midbrain of zebrafish, and on the left, we have a wild-type animal. On the right, we have a RAG-A mutant. And this is about the midline of the animal. And you will be seeing macrophages infiltrate from the lateral periphery that is shown by the arrows. And I'm going to start the movie. And in wild-type animals, you will see a large number of macrophages infiltrating over time and colonizing the brain. Whereas in Grmai mutants, I'm going to play the movie again. In Grmai mutants, very few of these macrophages enter, and even fewer stay on in the brain to form microglia. And this observation is quantified on the right. And since microglia are a kind of macrophage, I also wanted to look at peripheral macrophages in Grmai mutants. And here are the head, yoke, and tail regions of a wild-type animal. And we found that in essentially all the time points during which we examined the Grmai mutants, they had considerable numbers of peripheral macrophages. However, I think you can appreciate that these macrophages have a very abnormal shape. They are really amoeboid, which is very similar to what Kim observed in the case of microglia in Grmai mutants. And therefore, based on these observations, I conclude that microglia and macrophages in Grmai mutants display very similar defects in early development. So for the rest of my talk, I will either be using microglia or macrophages to examine the Grmai phenotype further, but I will be happy to discuss during the Q&A session as to how I distinguish between these two very similar cell types in these early stages of development when the blood-brain barrier is not fully formed. So in the first of these experiments, I isolated cells of the macrophage lineage from wild-type siblings and Grmai mutants, extracted RNA from the macrophages, and performed RNA sequencing. Gene ontology analysis of the differentially expressed genes, especially the upregulated genes, revealed an enrichment of categories pertaining to lysosomes, particularly these cysteine proteases called cathepsins, as well as endosomes and the proton-pumping ATPase complex. Now, since Grmai is a lysosomal GTPase, this differential expression of lysosomal genes was not particularly surprising. What was surprising, however, is that we found a significant downregulation of genes related to migration and chemotaxis, as well as immune response. And these functions of Grmai had not been appreciated previously. Now I will show you how I proceeded to perform in vivo validation of these transcriptomic observations, starting with the upregulation of the endolysosomal pathway in Grmai mutants. First, I generated a genetic construct in which genes encoding the lysosomal proteins LAMP1 or LAMP2 tagged with mCherry are driven using the MPEG promoter. And whereas in wild-type animals, we see some expression of LAMP1 and LAMP2, in Grmai mutants, these compartments are significantly expanded, and this observation is quantified here. I also generated labels for early and late endosomes by tagging RAB5 and RAB7. And once again, while we see some expression of RAB5 and RAB7 in wild-type animals, these early and late endosomal compartments are significantly expanded in Grmai mutants, and this observation is quantified here on the right. Another category of genes that was significantly upregulated were those belonging to the proton-pumping ATPase complex. And I performed functional validation by treating the zebrafish larvae with lysosensor green, which is a dye that specifically accumulates in acidic compartments of the cell. And you can see that the Grmai mutants show higher intensity of lysosensor green punctae in their dorsal midbrain, indicating that these animals show reduced pH of their lysosomes. Finally, degradation in lysosomes also requires the activity of proteolytic enzymes such as cathepsins and other proteases, which require low pH for their activity. And these the activity of these proteolytic enzymes can be assayed using dyes such as DQDSA or magic red cathepsin, which are dyes that are normally not fluorescent, as you can see in the wild-type animals. But when proteolytic enzymes cleave away the quenching residues of these dyes, they acquire bright fluorescence, as we see in the case of macrophages in Grmai mutants. And this observation is quantified here. So together, these observations confirm that indeed endolysosomal genes are upregulated in Grmai mutants. And perhaps at this point, you're asking yourself the question that we asked ourselves. Does this increased lysosomal activity in Grmai mutants really lead to more effective clearance of debris? And this is not merely an abstract idea, right? Because in many diseases, especially neurodegenerative disorders and lysosomal storage disorders, where misfolded proteins are observed, many groups are investigating the possibility of enhancing cellular clearance by activating the endolysosomal pathway and thus degrading the proteins and hoping for improved pathology of disease. So we wanted to take a closer look at the features of microglia and macrophages that are essential for debris clearance. And first, to clear extracellular debris, of course, microglia and macrophages need to be able to sense these particles and migrate towards the debris. However, we found that macrophages and RAGA mutants actually show a significant downregulation of genes related to migration and chemotaxis. Particularly, the CSF1 receptors are essential for macrophages to migrate and colonize their tissues. And we found that both the zebrafish homologs of the CSF1 receptors, CSF1RA and 1RB, were significantly downregulated in our RNA-seq. And to functionally investigate whether or not macrophages respond to chemotactic cues, I performed tail fin injury of the larvae followed by time-lapse imaging. And whereas in wild-type animals, the macrophages rapidly accumulate at the wound site, macrophages in RAGA mutants do not show such a directional movement. And in the beginning of my talk, I showed you that embryonic macrophages are in fact unable to migrate and colonize the brain. And combining these two observations, I conclude that macrophages in RAGA mutants do not respond appropriately to developmental or injury cues. Next, we wanted to look at the debris clearance functions of microglia and macrophages. And the easiest way to look at the clearance of apoptotic debris is, of course, by performing tunnel. And so I looked at whether neuronal debris in the brain is cleared effectively by microglia. And here you can see that microglia in both wild-type animals and RAGA mutants are filled with neuronal debris and shown here by these arrows. However, RAGA mutants have significantly more uncleared debris in the brain, and this observation is quantified here. However, if you recall, RAGA mutants have significantly fewer microglia. So of course, the reduced debris clearance could be a result of the fewer microglia in the brain. So what we wanted to do was, since there are considerably more macrophages in the periphery of RAGA mutants, we wanted to test whether macrophages are able to clear a different kind of debris. And so we looked at microbial debris, and E. coli, the primary function of macrophages is to clear microorganisms, other pathogens that could be invading. And we can test whether macrophages are able to respond to microbial debris by injecting E. coli tagged with Texas red. And here you can see that macrophages in wild-type animals rapidly take up E. coli particles, and in the process, they become activated and amoeboid. Whereas macrophages in RAGA mutants show very little co-localization with the E. coli particles. And consistent with this observation, we found that pathways related to immune signaling, inflammatory response, defense pathways are significantly suppressed in macrophages from RAGA mutants from our RNA-seq. So it appears that macrophage, in addition to not being able to sense developmental and injury cues, perhaps macrophages in RAGA mutants are also unable to sense these microbial cues. So in this part of my talk, I have shown you that even though there is an expansion of lysosomal compartments in RAGA mutants, this does not really lead to enhanced clearance of debris. Embryonic macrophages in RAGA mutants also do not respond appropriately to chemotactic cues. And finally, I have developed tools to visualize the endolysosomal pathway in microglia and macrophages. And I will be using these tools in my own independent lab in the projects that I will discuss next. So in the last few minutes of my presentation, I'd like to share some of the ideas that I intend to pursue in my independent lab. And the first of these projects is focused on these key lysosomal transcription factors called TFAB and TFE3. So I didn't get a chance to share this data today, but I have shown that RAGA and its partner folliculin repress these transcription factors for the normal development of microglia. And I also showed that TFAB and TFE3 specifically activate lysosomal pathways downstream of cellular stress. And the observation that cells have this dedicated conserved signaling pathway to turn on lysosomes in response to stress led me to design the first project that I will pursue in my own lab, where I will ask how are signals of extracellular stress transduced to TFAB and TFE3. Asking this question is important because a broad understanding of how macrophages respond to stress will be crucial to our understanding of how these key immune cells protect our body from all forms of injury and insult. And as I was wrapping up my projects on TFAB and TFE3, I also became aware that the Alzheimer's disease community was increasingly appreciating that many genes associated with Alzheimer's disease risk are enriched in microglia and function in lysosomal pathways. Genome-wide association studies from other neurodegenerative diseases do not show such a strong microglia signature. Simultaneously, I also realized that the functions of most of these genes in development and homeostasis were unknown. And understanding the normal functions of these genes can be key to understanding how these functions are disrupted in aging or disease. And I reasoned that zebrafish would be especially suited to probe the normal functions of these genes because A, zebrafish are particularly suited for large-scale functional analyses, and B, the feasibility of live in vivo imaging in zebrafish is especially valuable for the study of functions of microglia. And I also want to point out that similar large-scale functional screens of schizophrenia-related genes in zebrafish has led to valuable insight into the biology of the central nervous system. So the goal here would be to take genes associated with Alzheimer's disease risk loci, identify their zebrafish homologs, and perform a large-scale CRISPR screen in zebrafish to understand multiple aspects of microglia formation and function, probe endolysosomal signaling, and compare the crosstalk between nervous and immune systems. And I believe that this approach will reveal novel functions for Alzheimer's disease-related genes, as well as uncover important and interesting biology of microglia and macrophages. And with that, today I have shown you that a lysosomal GTPase is necessary for chemotaxis and immune function of embryonic macrophages and microglia. That experiments we will be doing in my future lab will reveal the signaling cascade that microglia and macrophages deploy to activate lysosomal pathways in response to stress. And finally, that I'm planning to do a functional genomic screen to illuminate the functions of Alzheimer's disease-associated genes to uncover interesting biology of microglia and macrophages. And with that, I'd like to thank members of the Talbot Group, especially my mentor, Will, as well as people who directly contributed to this project, my absolutely wonderful trainees who infused my lab and the work with their interest and enthusiasm. And finally, my truly generous funding sources, without whose support I would not be able to perform this work. And I will be happy to take questions.
Great. That was an excellent talk. Thank you.
Thank you.
I cannot wait to see how you move this work forward in your own lab. So we have time for a few questions. I'll start right off with one from Donn. He says, great talk, Harini. In zebrafish, I believe there are two populations of microglia, a transient primitive MIB-independent population derived from the yolk sac, and the other definitive MIB-dependent population derived from the hematopoietic stem cells. I wonder if you examined both sublineages, would you detect differences in lysosomal activity?
Yeah, that's a really great question. Yeah, you are correct. There are definitely two independent populations. One is the larval population of microglia, and then the other is one that's specified later, the adult population, starting at about 7 days post-fertilization. And so all the experiments that I have shown today, they are all from the larval stages. So they are between 4 and 5 days post-fertilization. So I was specifically looking at embryonic microglia for all my experiments. But you are right that it would be very interesting to look at the adult microglia and look at the requirement of not just TFAB and TFE3, but whether lysosomal pathways are similarly required in these cells that are specified later. My guess is, of course, lysosomal pathways will be required, but I think perhaps related to my project, the more interesting question is, do TFAB and TFE3 play a role? And we don't know the answer to that. And that would definitely be a very interesting question to follow up on. Thank you, Donn.
Great. We have a question from Louis Prahl, who asks, could the increased lysosomal activity mean that microglia are degrading proteins they need for migration, such as integrins? This might explain why they switched to amoeboid migration.
That's such a great question. I'm not sure. I don't, yes, of course it's possible, but I think there could be, so most of my data at the moment show that the microglia and macrophages are not really able to sense debris the way that they should. They're not able to migrate towards the debris. They're not able to eat it up. They're not able to digest it. But one possibility, I think, is that they could be misdirected, right? Like, so microglia and macrophages are presumably very specific with respect to the debris that they clear. And, you know, they could be eating the things that they are not supposed to. And yes, thus it is therefore possible that they are eating up the things that are actually helping them, such as migration and signaling molecules. So that's possible. I don't think anyone has tested that, but that would be a very interesting follow-up.
And we have a question from Kyle. Thanks, Harini. Are other cell types aberrant in the RAGA mutants? If not, why are myeloid cells such as macrophages, microglia uniquely vulnerable to lysosomal hyperactivation?
Thanks, Kyle. Yes, that's a great question. Yes, definitely. RAGA is required in other cell types in zebrafish. In fact, the RAGA mutants don't survive past 12 days post-fertilization. And although the macrophage phenotype is probably contributing to that early death, I think there are other cell types, cardiac, we know that RAGA functions in other cells, right? And yes, so that is definitely true. And I think that the reason macrophages and microglia are uniquely vulnerable to lysosomal hyperactivation could be because of what I mentioned earlier about this really important role of lysosomes for macrophages to be the cells that they are, which is professional phagocytes, right? And therefore, I think microglia and macrophages require lysosomes in a way that perhaps, I don't know, I don't want to throw a random cell under the bus, but other cells may not require as much, right? Yeah, great question.
And in the interest of time, I'll ask one more question that you can take a look. There are a couple more. A question from Enrique. He says, very interesting presentation. Calcium signals are important for the immune response. Do you know if calcium signals are dysregulated in the lysosomes of RAGA mutants?
We don't. I really do want to follow up on calcium signaling in my own lab, both in microglia and macrophages, as well as in other cell types. And I would love to talk to you more about it. Maybe we can get in touch. I just want to super quickly answer the two questions that are really easy. Augusto, yes, RAGA knockout siblings have a much shorter lifespan. They only live up to 12 days post-fertilization and then they die, and thus indicating the importance of lysosomes for the function of for just the survival of the animal. And yes, there are behavioral assays too for Alzheimer's disease. There are some really great ones. Matt Parker, who does a lot of these working memory and executive function assays, has a lot of these great assays. So yes, Donn, we can talk more about it if you're interested in these assays. Yeah, thank you all for the really great questions. Thank you, Lydia, for moderating.
Great, of course. Yeah, my pleasure. Yeah, so if there are any other questions, I would encourage you to reach out directly to the speakers after this talk ends. So thank you to both Daniel and Harini for your excellent talks. This seminar has been recorded and will be available on the SDB website next week. And please join us for next month's seminar on Friday, January 13th, when Dana Shaw from WashU and Joseph Campanale from UC Santa Barbara will present. And thank you all for coming.